Are you ready to get started with Science and Beers? Please give a very warm Friday night welcome to Nathalie Henriksen, everybody.
Thank you, Michael, and thank you all for being here for the microbes and the beer. But I'm just gonna get started because now we already talked a little bit about microbes. And I'm gonna start talking about the title because some people already talked about this, and also the other day when I told a friend I was going here and she said, "What is the title of your talk?" And I was like, "Oh, it's super cool. It's like microbes on the microphone." And she was like, "That's disgusting." And then I realized I have not thought about that because I actually rarely think about microbes being disgusting. Because the way I work with them is in a beneficial way. In the good way, I always look at them as my best friends, as the ones that's gonna help us actually do a lot of the things that we need help with. So the meaning with this title was not that we're gonna talk about how disgusting things are and how microbes can make us sick. There might be a little bit about that. But actually, it's gonna be about that microbes, they also have a voice. But we can't really hear it. So tonight, I'm gonna try to get that amplified. I'm gonna try to be the one speaking for the microbes and tell you about what they are actually saying. So to get a little bit away from this disgusting thing with microbes being on stuff and we don't get disgusted by that, I thought let's start with something cute instead. And I'm gonna start with this little guy. It's a squid, for the ones that doesn't know it. It's called a bobtail squid. And this one is from Hawaii, actually. And it's not very big. It's on like the size of your fingernail, making it even more cute, yeah, I know. And it lives in these super clear waters in Hawaii, tropic. And it can't really go out during the day, because during the day you have the sun and it's shining and it will reveal it. So predators will come and hunt it, and it can't really hunt its own prey. So during the day it hides. And it can look something like this. This is actually not the Hawaiian one, I have to say. It's disclaimer. This one is living another place in the tropics, but it's exactly the same one. So during the day, it does something like this. But then when it becomes night and it needs to go out and hunt, it still has a problem. Because in Hawaii, the water is so clear that the moon, when it's shining, is still revealing it. It will cast a shadow on the bottom. So what does it do? Well, it needs some kind of camouflage. And what is the best camouflage when it's the moonlight that is showing you off? Light. So this little squid has underneath it, it has an organ that we call the light organ. It's a very small thing in its mantle, so in its like cape that it has around its body. And in there, it produces a light. And it's so bright, it's called counter-illumination, that technique. So when it goes over the sea bottom, well, things underneath can't see because it looks like the moon. And it doesn't cast a shadow, so things from above can't really see as well that it's having a shadow. That's pretty clever. But what is more clever is it's not itself producing it. That is bacteria. So inside this small organ, there's a huge bunch of bacteria. These bacteria, they're producing this light. And to begin with, when researchers many years ago discovered this, they were like, this is pretty weird. Because the bacteria that lives inside the squid, they also live in the water around the squid. And there, they don't produce any light. So they do it when they're inside, but not outside. What can be the difference? Well, what they found out, and I promise you, this is the only graph you will see tonight. And it is super simple, but I will still explain it. So here on the x-axis, we have the amount of bacteria. So when we go to the right, we get more bacteria. What the researchers saw when they were studying this in the lab was, as the density increased, there came more bacteria. Suddenly, they hit a threshold, and then they start illuminating. So they didn't do it when there were few, they did it when there were many. The thing that controlled this was a signal molecule. So they saw that they had a molecule that needed to increase in amount. And when they hit something, then all the bacteria together said, let's go, guys. And then they started illuminating. And this signal molecule we call an autoinducer. But let's also just call it, it's a way of talking. Because what they use it for, or what researchers say they use it for, is to communicate and tell we are enough. It's a way to make bacteria see each other. Because now, at least for now, we think bacteria they can't see. So they are blind, so how can they tell that they are enough? So this is a way they can sense each other. This kind of communication we also call quorum sensing. You don't have to remember that term. And quorum sensing they actually use for a lot of things. Not only to make light, but also to coordinate other behaviors that they have to do together. Because one might ask, so why do they have to do it when there are many? Why not a few? Is there any beneficial in being a population that's doing it at a specific time? And yes, there is. For example, this kind of communication, it's also happening when sometimes pathogenic bacteria, so bacteria that makes us sick, when they, bless you, not that kind of sick, it's actually even more worse. There's a bacteria called Pseudomonas aeruginosa. It can make people really, really sick. When it infects a person, to begin with, it doesn't really make any virulence factors, so what we can, toxins and things that makes us sick. But at some point, they start making it, and that's again when they are enough. And they do that because if a single one started producing toxins alone, our immune system would sense it and say, oh, there's some bad guys in here, let's kill them off. That's not really good if you are pathogenic bacteria. Not that they want us to be sick, but that's what's happening. So what they do is they go in stealth mode. So they stay below a certain population size, and when they are enough, then they know if we are kicking it off now with all our toxins, then the human immune system will not have a chance. And then they start kicking off, so they have to decide or coordinate when they are enough, and that's what they use these communication systems for. I did actually bring a very complicated figure, and I will now go through it step by step. I will not, because I can't actually remember all of the steps, but what I wanted to say is, within each cell, it's not this communication molecule, this autoinducer that is making a light, but this one is sensed by a system that is inside the bacteria, and then this system starts and then produces this light. All in all, what is going on here is a very beautiful symbiosis. So we have a squid and a bacteria that lives together. So the squid gets help to go out hunting because it can't be seen. And what the bacteria gets is a nice place to stay. Because it's only this bacteria that lives inside this squid. And in there, it's a party. There's all the things you want as a bacteria. There's all the nutrients that you want. There's no other ones to compete with. And you can stay there almost forever. Because during the day, the squid doesn't want these bacteria to illuminate, because then it will be seen by everyone. So actually in the morning, the squid expels all of them. It casts them away. But there's only a few of them left still inside this light organ. So then during the day, these bacteria, they grow, they double, and then they hit this threshold around evening again, and then start illuminating again. And this cycle goes over and over and over. And that is, of course, something you can be like, wow, okay, that's quite amazing. There might have taken some time for them to figure this out. And yes, you're completely right. So let's just take a trip down memory lane and think about when bacteria and small microbes, they actually came to Earth. So let's start with us. That's a good place to start. We came around humans around these 2 million years ago. And then a lot of things have happened before that, as you can see. And actually all these very complex animals, they are really old, but not as old as bacteria that I'm up here is calling prokaryotes. So does everyone know what I mean when I say microorganisms? Is there have you heard the terms bacteria, virus, microalgae? See some nodding. I'm still going to say microorganisms, which is a word that I use a lot. It's actually it's a weird definition because there's no set in stone what is a microorganism. What we normally say, it's stuff you can't see with your eyes. That's how biology works. We don't really have any clear definitions in that way. We try to have it, but it's really difficult to have anything in biology contained in a box. So microorganisms can be bacteria. Then we call them normally prokaryotes. That's the way their cell structure is built. But we do also have microorganisms that have the same kind of cell as the ones that we have. So we consist of a lot of cells, but sometimes single cells in the same structure as ours also exist. For example, microalgae. And we actually are able to see microalgae sometimes. Now it's a little bright in here, but maybe you can see a little bit, there's some dolphins jumping around here in the water. And it looks a little bit magical, a little bit like a Harry Potter world, but this is actually real. Have any of you experienced ever being out, some people out in the waters when there is in Danish what we call morild? Yeah, so sometimes during, especially now, it's a little late now, but in Denmark during the late summer, we have these huge blooms of dinoflagellate. So that is kind of a microalgae. And they do the same kind of illumination as the bacteria do. So this with illumination, it actually occurs all over the place with different kind of animals. Most often it's microorganisms, but there's also some animals that can do it themselves. But when dinoflagellates, these microorganisms, they illuminate out in nature, we're actually able to see them. Researchers are not really sure why they do it. Is it communication as well? Is it a way to protect themselves for sure? They're not sure. And I'm not going to go into that because that is not my research area. But there's also other places where we are able to see these huge densities of microorganisms. So I went into the European Space Agency satellite pictures to find some of these pictures of huge blooms of microorganisms. Here we have something on the north coast of Norway, you might see. So these milky white kind of densities, pretty normal, they occur. We have these blooms all over. And I'm pretty sure many of you normally know microorganisms or microalgae as being something annoying. It closes down the beach because there's too many and it's toxic and then you can't go swim and it's super annoying. But there's also actually a really good thing about microorganisms, or especially microalgae. And
take a breath. Now take another. Now think about this. One of those breaths was entirely provided by those little fellas under the sea.
Couldn't have said it better myself. You always need Will Smith to give you a great point. So what he is saying is actually these microalgae that we have around in the seas, they are actually producing the oxygen that we breathe. They are actually responsible for creating the atmosphere that we have today. And it is half of the oxygen we have in the atmosphere that microalgae they produce. So they're quite important. And it's quite important we have a lot of them. Not always in a bloom, but it's really, really important. And let's just go back to one of the kind of microalgae again. So now it doesn't say on this picture that I downloaded what kind of microalgae. They are actually capable of analyzing that in the space agency, because based on the kind of reflection and color it has, they can actually say, oh, it's specifically this type. But it didn't say, but I have a quite good guess what this kind of microalgae is, because it's milky white. And there's a special kind of microalgae that appears milky white when it's in huge densities. And that is something that is called in Danish, it's called a kelpflagellate. In English, it's called a coccolithos. And I'm just going to call this one Emiliana Huxley. This is a very, very beautiful microalgae. This is a really fancy picture that you can take of microorganisms. It's taken with electrons. So when you want to look at something so small that you can't see it with your own eyes, and you want to have all the beautiful structures, you use these kind of pictures. What you see here is a single microalgae. And it's covered in these shells, and they're made out of something that is similar to limestone, so kelp. They are actually also responsible for making a lot of the sediments we have around in the world. You can think about when you look at that. What is quite special about this is that it's really responsible for a lot of the carbon cycling we have out in the oceans. These ones are so active because they do photosynthesis. So they live, the sun is the energy and CO2 that almost all living organism is producing. They use that as an energy source. And then they build it into their body mass, and then when they die, it sinks out or they get eaten by something. They are really productive and really important in our ecosystems. What you should know about this is there are some things that controls whether they die or whether they live. And that has something to do with bacteria. So these guys are not living alone, of course. They have a lot of bacteria living around them, just like us. They have a microbiome inside them. And what happens is when they're out there happy and growing, they produce molecules that is called DMSP. Let's just call it that. And that is a molecule that is delicious for bacteria. It's full of carbon, it's full of sulfur, all the stuff that bacteria needs to grow. So it attracts bacteria, especially a bacteria called Pheobacter inhibans. Pheobacter inhibans get so excited about this molecule that it starts producing some other molecules that this guy is really happy about. It helps it growing. It's another kind of nutrient for this. So that's super beneficial. But the Pheobacter also want this algae for itself. So it also produces another molecule, which is brown when you look at it in a laboratory. Come on, and it's there. And that one we call tropodithioidic acid, short TDA. TDA is a signal molecule. It's just like the chrome sensing molecule I showed you in the beginning. Pheobacter uses it to communicate with each other, to decide, now let's go down here, guys, and settle down and build a home around us, which we call a biofilm. But it can also kill off specific bacteria, bacteria that are not beneficial for the microalgae. So this works as an antimicrobial compound as well. So it has multiple roles. This is a great relationship. But then forgot. I promised my PI that I always show the structure of TDA whenever I'm presenting it. Now you've seen it, let's move on. She's gonna be there in Copenhagen on Wednesday. I'm gonna spend a little more time on it there. Yeah. Okay, at some point, this microalgae will get old, like everything. So it starts to release some compounds that are different. Actually, some kind of lignin-like molecules, that's the same we have in trees. And when they secrete this, Pheobacter senses it. And then it shifts its mode. It stops producing these beneficial compounds. And instead, it turns into this reddish compound called rosseobactericides. And yes, they kill off the microalgae. So suddenly it goes from being its best friend to suddenly being the one that kills it. And why would you do that? Well, you will do that because this microalgae is done. Everyone is going to have a feast on it in a minute. Why won't you just be the one getting there first to get all the delicious nutrients inside it? So this bacteria is living so close, and then it's the first one to kill it and to get everything. That's a pretty beautiful technique to actually have out in nature. So this is a very complicated relationship, and it's controlled by all these molecules because rosseobactericides, they're also a signaling molecule. Other bacteria or the same kind of bacteria can sense it and know then, oh, there's a feast going over, let's get over there and get into it. So one thing in this relationship I have been specifically interested in, and that is TDA. Because we do know that it can kill off some bacteria and that the Pheobacter bacteria that produces it, they can sense it. but we're actually not sure who else can actually understand this compound. It is a signaling compound. So one of the first things I did during my PhD was actually to try to look into who else can understand this compound. Is there someone else listening in on this? And the way I did that is can be explained in 3 minutes. And I did that on the television show, so you can go home and see that. And I will try and make it a little bit shorter than 3 minutes, because simply what I did was I took this kind of microalgae, and then I have it in different containers, and then I added in this bacteria that can produce the compound. And then I had some other containers with the microalgae with the same bacteria, but then I had modified it, or someone else actually modified it, so it couldn't produce the molecule. That's quite important, because that shows me when I add in someone that can produce the molecule, then the ones that will change the behavior down there, that will be based on the molecule, because in the other one, the molecule is not present, it's only the bacteria. So it's kind of a control. I did this because I wanted to see what kind of bacteria, together with this microalgae, is behaving differently. I did this over 2 months, 3 months, I think. I had this experiment going on, sampling, looking at who is there, who is not there, crunching data, sequencing DNA. I did a lot of stuff, and I looked into all the data afterwards. And I can say there's around 60 different kind of bacteria living together with this algae. And then when I saw my final results, I found out that one was actually responding. And the crowd goes wild. Yes. Is it good? Is it bad? That is a little difficult. I can tell, yes, not super exciting when I found out, because it could have been nice if everyone was like changing their behavior and was responding to this, and I could be like, oh, this is the master compound of the world. Everyone is responding. And there was one single fella in there that responded. And there was a bacteria that looks very much like the one I added. So it's kind of its friend just responding to it. And in that way, you could say that's kind of logic, maybe, because they look so much alike, so they probably understand each other. But actually, it is something that is quite important. Because maybe you are saying now, and so what? What can we use this kind of things for? Guys, let me tell you. Aquaculture, of course. Yeah? Do you know what aquaculture is? Fish farming. Something that looks like this out in the world. So we have a lot of fish we have in these containers out in the world. We grow them so we can get a lot of food. Actually, this is a growing industry. If you look over time and you see the million of tons of fish that we eat in the world, you can see that we used to capture a lot of fish, but now we're actually farming more fish because we have a growing population. This is a huge industry, and it has, of course, some huge challenges. Because the small fish larvae that they rear in these big containers, they get sick sometimes. And when they get sick, they have a problem. Because you can't vaccinate a fish larvae. They don't have an immune system. So you need to treat them with medicals. That's not really a sustainable way, because you throw in a lot of chemicals and medicines and antibiotics. And that can be a lot of troubles with that. Let me put it in that way. So what we found out is actually this bacteria, Pheobacter, that produces TDA. TDA can kill off some of these fish pathogens. So now there's companies like, it's called Lallemont, that looks into using this bacteria alive, so as a probiotic, add it into the fish rearing system or the larvae system with the live feed that they get, and in that way protecting the fish larvae. So where is it my research matters? Well, it's kind of good that we know not a lot of the other bacteria cares. They don't care if TDA is produced. They are just going on with business as usual. It's only the fish pathogens that will be killed off. So that's quite good news. And speaking of antibiotics, that's going to be the next thing I'm going to dive a little bit into, because antibiotics, they are antimicrobial. But first, let's have a beer break. I just have to say, it took me 2 years to learn how to pronounce that name. So I'm quite impressed if you haven't heard it before. Have you heard it before? Fair. But still, good job. And TDA, that was a tropodithetic acid. That took me 3 years to learn how to pronounce. Okay, we're back. I actually got a question during the break from up here if I'm going to talk about viruses. And no, I'm not going to talk about viruses, unfortunately. Viruses is also, okay, I'm going to talk a little bit. They're also a part of this big blob of microorganisms. They're even smaller than bacteria and microalgae. And they are controlling, you could put it in this way. You have microalgae that controls our world to some extent. Then you have bacteria that controls microalgae, and then you have viruses that control bacteria. So that is kind of the hierarchy that we have. One funny thing about viruses, I don't know if it's funny, but it's good knowledge. It's actually, in some places in the science world or between researchers, there's a debate whether you can actually say that viruses is life. And that sounds weird, but the thing is actually, just with us, bacteria, microalgae, elephants, all kinds of animals, we are capable of reproducing ourselves without help from anyone. So that with that I mean our cells, they can copy themselves and make a new cell. Viruses, like a coronavirus, they can't actually. They need another cell to do that. And that makes some researchers actually debate, is it then life? If it really can't replicate itself, that it needs to hijack someone, use their systems in order to copy their own DNA. End of that debate, because there's a lot into that, but that's kind of the only thing that I talk about with viruses. Because I'm going to go back to the last thing I said, where I said antibiotics. It's going to be a little bit about antibiotics. So here's the deal. TDA is an antimicrobial, but you could also say it's an antibiotic. Because antibiotics, that is originally produced by microorganisms. Nowadays, we do a lot of synthetic compounds that is then antibiotics, but actually antibiotics is something we found in nature. And I'm going to take you a little bit down how that happened. We're going to go back in time. And back in 1940, this fella in Oxford, he was an officer, and I can't remember his name. He got a scratch from a bush of roses or something like that. He was doing some garden work, and then he got a scratch in his face. And that got infected with some bacteria. And at that time, that could be super, super dangerous, because there was no cure when you got infected with bacteria that went into your bloodstream, for example. But luckily for this guy, a couple of years earlier, okay, a little more than that, in 1928, oh yeah, okay, blah, 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 antibiotics. This guy, Sir Alexander Fleming, a Scottish microbiologist, he had an accident in his lab. And the accident was that in this petri dish, this is a way that we grow bacteria or microorganisms in the lab. We have a small plate, and in that we put all the things that microbes they like in like a jelly kind of solution, and then they grow on top of that, and then you can do all kind of things with them. He had some bacteria growing in that, some pathogens, some that can make us sick. And then by an accident, some stuff from around the room fell down on it that he couldn't see, but at one point it grew up. And he could see, oh, this is actually a fungi, which is also microorganisms. And he could see where it was growing, the bacteria was not growing. So he knew there's something that this fungi is producing that the bacteria doesn't like. And he did find out this is a compound it's an antibiotic. But he could not really get it purified, so get the compound alone. So he couldn't really treat anyone with it or try it out on humans. Fast forward to 1939. There were some other researchers that were actually capable of purifying this antibiotic, and it's called penicillin. And these researchers, they were able to produce a tiny, or purify a tiny amount of this antibiotic. And that one, they tried on the officer that I showed you. And he actually got cured for 11 days. But then they ran out of antibiotics. And then he died, unfortunately. Okay, that was not really happy, sorry, but in the name of science, what actually then happened, okay, we're going to take a sidetrack here, is they knew, okay, this is a miracle compound, like you can cure people that gets infected with bacteria, but we need a lot more. And then, you know, in the 1940, the war broke out in Europe. This is happening in Europe. And these scientists that tried to purify it, they knew this is a miracle drug, and we don't want that to get in the hands of the Nazis. So what do? We need to get it out of Europe. So they called America and said, hello, America. We have this thing, and we think this is gonna save the world. Can we get it to you, and you can help us purifying it? And America said, yes, let's do that, but you need to like smuggle it out of Europe, because in case some Germans find you, they can find this fungi that you have growing on the plates, and then they will start producing antibiotics and can cure all their soldiers. That's not good. So what the researchers actually did was they had some coats on, and then they took this fungi, which produces, what fungi do is they can go in this kind of hibernation state, where they turn into a small, we call it a spore, so kind of a seed, you can say, where they're not really alive, so they just go in hibernation, and then when they have favorable conditions again, they can bloom up again and live on. So what they did, these researchers, they took these spores and then they put it on the inside of their coat. Because as you know from your own coat, sometimes it smells a little bad and you say, ooh, there's something, oh, it needs to wash it. That's normally because the fungi we have growing on our skin gets into our clothes, and especially, you know, your training clothes, it smells really bad. That's fungi. And maybe your washing machine, it also smells really earthy in some way. That's also fungi. So they took it and put it in the coat, because there they could live on it. And then they could go to America, and then they could take their coat and put it on petri dishes, and then they can grow up the fungi. That's how you do it. There's a great book about this story. The other great thing is actually, in the States, they were capable of purifying enough that they could treat people. And to begin with, they only treated soldiers. It was not something for the public. And right after the Manhattan Project, antibiotics was actually the most funded project in the States. It was so valuable for them, because during World War I, yeah, there was a lot of soldiers that died due to getting shot, but what they actually, most of them died from was infections that they couldn't be cured for. So they knew if we get this compound out to the soldiers, we can actually save a lot of them when they are in war. And then after, when the war ended, it got released to the public. It actually looks something like that. Around in the States, they have these kind of posters talking about how this was a miracle. So it's actually quite fascinating. One thing then that happened after this discovery of penicillin is actually that we discovered a lot more antibiotics. Because researchers started going out and say, well, if this fungi can produce something that can kill bacteria, there might be other drugs out there or compounds that is mass weapon destruction kind of thing. So during the years, they started discovering many kinds of different antibiotics produced by microbes. But at the same time, resistance also started showing up. This is resistant bacteria. So we started treating people for bacterial infections, but then at some point, some of these bacteria, we couldn't get rid of them again. They were actually resistant towards it. So we have had this kind of race going on between resistance and discovering new antibiotics. And in the 1990s, we discovered the last antibiotic. Ever since that, we've only had new resistant bacteria occurring. A lot of abbreviation, I can't remember all of them actually, but it's a lot of different kind of E. coli and Staphylococcus that is resistant to all the drugs. Actually, during the COVID pandemic, so from 2019 to 2022, around 5.9 million people died of COVID. But alone in 2019, 5 million people died due to antibiotic resistance. This is a huge problem in our world, that we have antibiotic resistance rising, and we don't have any new drugs. Thank you. No. It is a problem, and researchers are trying to look into what do we do? One thing that we have realized is resistance is occurring much faster than it's happening out in nature by itself. Because resistance is like a natural way that bacteria respond to something that is threatening their life. So they are getting treated with these weapons of mass destruction, and then of course they evolve into something that can resist it. That's natural, but it's happening too fast. And it is that because we use antibiotics in so high concentrations. For example, if you're treating a patient for tuberculosis, we use, I think it's 160 times higher concentration of antibiotics than is needed to kill the bacteria that is causing the tuberculosis. And there's a good reason we do that, because we want to make sure that we survive. This is how we treat, so we can get fast over patients as sick, of course. But it's also stressing a lot and making this resistance happening faster. Another thing we have noticed is when you look out in nature, you never see any microorganism producing antibiotics in so high concentrations. It's not natural. They produce it in much lower concentration. And when they do that, the antibiotics is actually having some other roles than killing bacteria. They actually work as communication, like TDA. So you can say everything will get toxic if you have enough of it. It's also with alcohol, you know, if you drink. Do you know that? Let me tell you guys, if you drink too much alcohol, that is also toxic. That's the same with any compound. If we get enough of it, it will be toxic. So what we have tried to look into is actually, well, if it's never produced in the amounts needed to kill bacteria, what is it actually used for in nature? That requires that we go out in nature and try to find the molecules and detect them at the levels that they are produced. And good luck to us, because that's what I spent 4 years on, trying to find one compound. So let me put it in this way. I did one experiment with a colleague, and it was an experiment where we wanted to look into a community of wild bacteria living in the ocean. So we wanted to look at this composition of bacteria, how they live together, and then we wanted to see what kind of signaling molecules and antibiotics are they producing. And we did that over 4 months. We went out in nature, we built some system ourselves, we did a lot of work, and this is in real time, guys. It's a lot of work. We collected a lot of bacteria, and it was the same place we went always to look, and we extracted DNA, and we extracted the molecules, and we crunched data again. God, we worked a lot. And we found around 3,000 different bacteria in this system over 4 months. That's a lot of different kind of bacteria. So that's just species. There was a lot of, but it was different species. And there was even more genes that we could see in our DNA codes, genes encoding these kind of signals. So me and my colleague Pernille, that is also working up here a lot and doing a lot of graphs over here, we 3 months before we handed in our Ph.D, we came to our PI and said, we have all this data, and it shows that there might be some molecules present. We can see the genes are there that is necessary for them in order to produce. Can we go and try some of these fancy machines we have and detect these molecules? And our PI said, no, There's 3 months until you hand in your Ph.D. Go home, sit down, write your Ph.D. And we went out from that meeting and we looked at each other and we said, let's go analyze these samples for the molecules. So we went to one of the people we have in our center that is really good at analyzing these molecules. And we took all our samples, like 200 different kind of samples from the 4 months, gave it to him and said, go crazy, let's find some molecules. And he crunched it all and looked through it, and then he came back and he said, there's one molecule. And you go crazy again, because this is really nice. Yeah, guys, it worked. Oh, such a struggle. So the thing here is, these molecules, signal molecules and antibiotics, they are super tiny. And they're produced in so low concentrations that it is so difficult to detect them. It's a little bit like, oh yeah, it was this molecule, by the way. It's called pseudanes. Pseudanes is known to coordinate populations of bacteria. It can also control microalgae. It seems like you heard the story before. It's a communication molecule. Super excited, but what I wanted to say is to look for these kind of microbial compounds is like looking at a reef full of different species, or you go to Africa and you look at there's lions and giraffes and elephants and all these animals, and then you hear one sound and then you're like, who said that? And you can't find out. That's how my life has been forever now. That we can sometimes hear a small conversation going on, but we can't really point at who is it, who said that. And then sometimes we can see that there's some bacteria present that is capable of actually talking, but then we can't hear that they're saying anything. So it's always this kind of puzzle trying to make everything work together. That was a kind of a hard study, but it was really good because we rarely detect any compounds in natural concentration in nature. That's also the case with TDA. So the study I told about in the beginning, where I added this bacteria, Fuibacter, that can produce TDA to these microalgae cultures, the truth is I never detected the compound. I know that it produced it, but I couldn't measure it. And that was under semi-artificial conditions because it was not out in nature. I took the microalgae in the lab. I had a lot of it. I had a lot of the bacteria, so it was kind of unnatural concentrations of these kind of microorganisms and compounds. So imagine then going out in nature where there's less of this bacteria, less of the microalgae, less of the compound. How on earth are we going to be able to detect that ever? Anyways, that's what I also tried, because some previous studies that someone showed to me said, we might have a feeling that this bacteria, Fuibacter, it lives in really high concentration in one specific kind of animal that we have also here in Denmark. And that animal is called moss animals. It's not a plant. It is an animal. It's a weird name. It's also called mosdyr in Danish. It's also called bryozoans, more scientifically. And many people don't know what moss animals is, and I completely understand that. But they are everywhere. So if you ever go along the beach and you find some seaweed, it can just be the very normal kind of seaweed that you see, or bigger ones like the sugar kelp that we also have around. You can see these white things growing on it. And you might think, oh, that's because it's dying or it's disease. But no, that is moss animals. So moss animals looks like this when you're up close. It looks like a coral if you ever looked at them. This works. One tiny one here, a little ring you see, is one animal. But it grows in these colonies, and that's why we can see them by our naked eye. They're not corals, they are their own phyla, which is a way to put everything into boxes. And these moss animals, they live a little bit like mussels and other filter feeders. So this is in a microscope. This is when you get really close and look at one animal. They have these lobophores coming out, and then they filtrate the water. And when they do that, they also take up microalgae. So the small green things you see here, that's inside the moss animals, and the green thing is microalgae. And alongside microalgae, we have Pheobacter. So this is an environment that I started thinking, could be ideal if we have a lot of microalgae in there. It's really concentrated. We have a lot of Pheobacter as well. There might be a lot of TDA. So maybe I'm going to be able to detect it here. So I took a lot of moss animals and I crunched it and I gave it to the chemistry person again. I said, go detect. And he came back and said, I can't detect anything. What do you do? I went to Germany. Because in Germany, at an institute down there, they are really, really specialized in a special technique called mass spectrometry imaging. That is a technique where you can take pictures of stuff. So you get like the spatial localization of stuff, and then it detects molecules. So it's the exact same mechanisms that we use to try to detect the molecules, but now we just take a picture with this kind of machine. And I can't explain, it's going to take 2 hours for me to explain how it works. So that's how I'm going to explain it. But the crazy thing is, what you need to do is you need to have small slices of the thing that you want to have pictures of. That means that I needed to take this bryozoan and do the same as they did in Star Wars, where Han Solo, he gets frozen. Have you seen that episode, some of you? The ones that haven't. At one point, Han Solo, he gets frozen and stiff like this in, I don't know what kind of compound they did that in. I do it in something called CMC matrix. And then they can keep him there forever, and then they can also make him live up again at some point. I don't do that with the bryozoans or the moss animals. What I do is, when they are encapsulated in this matrix, I can do tiny, tiny slices. And it looks like this. So this blob you have here, that is a capsule with my bryozoan. And then I have a very sharp blade, and then I can make a tiny, tiny slice. It's micrometer size. So now I have a slice of bryozoan. This is really nice because this instrument that is taking the picture, it's really sensitive. It doesn't need a lot of it. It just needs to go over the sample, and then it will shoot some lasers at it, and all molecules in the world will reflect at some kind of wavelengths, and then this camera can detect it. So that's what we do. We put it into the machine, and then it runs out, shoot a lot of lasers on it, reflects back, give tons of data saying it could be this compound, this compound, this compound, and this compound, and not this compound, and it does a lot. And then we can get these kind of pictures. So this is the bryozoan. The blue color is just some kind of a compound that is, or molecule that is all over a bryozoan. You can say it's a protein, or it's a lipid, so some kind of a fatty molecule that it consists of. And lo and behold, one place up here, if we zoomed in, there was something reflecting at another wavelength. That might be TDA. And I can't say more than that because it's actually not done, that study. That's quite exciting because we've never been able to detect it in nature. What I'm trying to say with this is it's super difficult to try to listen in on microbial conversation out in nature. And we need these kind of instrument that is actually, they are normally used in the world of medicines because you use it to do a lot of samples of human tissues, and then you look at where have we different molecules going on. But now we're starting applying it in microbiology or in the biology world of animals. And that's a little bit how it works in my world. We look out in other research fields and like, what kind of techniques do they use? Could we apply it in microbiology? So that's what we're trying right now. This is like, we are on the edge of what is capable, and we are there because we now have some technologies that can help us. And to be continued, because I'm actually handing this project over to a new PhD student in the center, because I'm continuing with other stuff. So he's gonna continue on this, trying to make us even more sure that it's actually TDA that we detect in these moss animals. And now you might think, oh, will it always be that difficult to study conversations and measuring bacterial conversations out in nature? And actually, no, because can you imagine the smell when you walk out in the forest and it's just been raining? Mm-hmm. Like that really fresh, earthy smell, where it's like, oh, there's some wet soil somewhere. A lot of people normally say when it's been raining in the summer, it's like, we love that smell. It's a really nice smell of this freshness. That is a bacterial conversation going on. Because what you can smell is called geosmin. Geosmin is a small terpene. It's a small molecule that a bacteria called Streptomyces, they produce. They live in the soil. And they produce it in, actually don't know in high concentration, but I know that we are super sensitive to it. We are really good at sensing it. Other organisms is also really good at sensing it. For example, earthworms, regnworme. They get, when they can sense this, they start moving a lot more. They're actually not at favor of it, but they move a lot more. Springtails, which is also a really small animal that lives in the soil, they get attracted by this molecule. This is because, or what researchers believe is, Streptomyces bacteria are producing it to attract springtails, because then they come by, and then the bacteria can hang on to the springtail that then jumps along, and then this bacteria gets spread out in the environment. The earthworms, on the other hand, they can eat Streptomyces. So when they sense it, the earthworms, they sense it as toxic, and then they go away, and they move really fast. That's quite beneficial, actually, that they start moving fast, because that's really good for our soil environment. And that's actually something that I've been quite interested lately. And also I know a lot of other people in Denmark is quite interested in our soil environment, because we are in these days in Denmark talking a lot about our way of using the land. Almost 2/3 of the Danish land is used by agriculture. It's almost, 32% of all land use in the world is agriculture. And the thing is, the way that we've been mostly using this land has been in a way that is not beneficial for the microbes living in it. But we are starting realizing, thankfully to technology investments, and that we have researchers that have looked into and figured out there's actually a lot of things going on in the soil, and the plants, they depend on it. You might have heard about these crises that we have normally when you have a field. A limiting factor for the plants is nitrogen. Nitrogen is something that all organisms needs. So we add it to the plants because that helps them grow faster. But we might add a little too much. And then what happens is that the plants can't take up all of it. And then it runs off down to the fjords, and then it makes everything else grow, and then that needs to be broken down, and then everything dies, and yada yada. That's a long story. The thing here is it's problematic, because we actually don't have to do that. We don't have to add it artificially, this nitrogen, because bacteria can do it. So this is some roots, and you see these big blobs on it. They are called nodules. It is a small environment full of bacteria that is capable of giving nitrogen to the plant. Because the funny thing is, nitrogen is not limited in our world, because in the atmosphere, over 70% of our atmosphere is nitrogen. But the plants, they can't take it up because it's in a form that they can't take up. It's because there's some really tight bonds in this molecule, and they can't break it down and then build it into their biomass. But bacteria, they can take this atmospheric nitrogen, And then they can make it into some organic nitrogen that the plant then can take up. And it happens all the time out in nature, especially in like peas and soybeans. They have these relationships where these nitrogen fixating bacteria, they live. And then they provide the nitrogen that the plant needs. And then you actually don't need to give them any nitrogen. So I know, and you can always go home and read a lot more about this. There is some initiatives that looks into this, looking a lot more of what is actually going on in the soil, what kind of microorganism is actually beneficial and natural for the plants to help them grow in a natural way. Where I'm coming into the picture is, well, this kind of nitrogen fixation is also coordinated by communication between bacteria and the plant, and bacteria between. Because actually, what the plant is doing is, when it's growing in some soil and there's not enough nitrogen for it, it sends out some molecules, some signal molecules, that these bacteria that can fixate nitrogen, they sense, so they get attracted, and then they sit, and then they start building this nodule and fixating nitrogen. But there's not a lot of research going on in this. But hopefully there's going to be a lot more, because this is very personal, of course. I think there's a lot of good ways that there's a good solution in looking into microbes, what they do, and understanding them in order for us to actually treat our soil and maybe even our waters in different ways if you understand what is going on in there. Oh yeah, actually now the number came. It's actually 38% of all the land we have in the world that is used for agriculture. And what is really, I read a report the other day about farming in Denmark and what we should do. And only one place out of 80 pages, they said microbiology. And it's so weird for me as a microbiologist when I know everything depends so much on microorganisms. If we remove the microorganisms, plants couldn't survive. It's the same with us. We can't survive without our microbes. So what I really hope is that we all start to realize that microbes, we need them, and we need to protect them in the same way that we protect other species. And that can be quite difficult because, as you can hear, I spent 4 years on just trying to detect one molecule in order to try and understand a bacterial conversation that is really difficult for maybe the broader Danish audience public to understand what do we need this conversation for. And as a researcher, I realized it is actually super difficult to tell you guys why I should research in this and tell you this is important for all of us. But during Forskerfesten, so the Ph.D. club I was part of, I actually started realizing, well, maybe, and I think this is something I said to a journalist when I was super drunk, actually, but it's actually quite good, I have to say it myself, is that we also need, as researchers, to go out and tell you about the process in science and research, how long time things take. I do have some time. Actually, I can try to point out it. Do you know the names Weissman and Carolina Carioca? Do you know the winners that won the Nobel Prize in medicine in 2023? No, I didn't either. Do you know the name Søren Brostrøm? Yeah, some is nodding. Do you do you know the names Moderna and Pfizer and so Corona? You know, the Corona vaccines, they came super fast. We put a lot in the world, a lot of money into research when Corona pandemics occurred, and we wanted to have some vaccines. And actually, it was quite amazing how fast we got some vaccines that were working. One of the reasons that we could that was because back in 2005, 2 researchers, they have been looking into how these kind of vaccines that is based on something called mRNA, how that affects our immune system. And they did research and no one believed in them and they couldn't get any more money, but they did figure out, if we put it in, this is how the immune system responds. Fast forward to 2019, because they did that research and spent years on that, we knew how we're gonna be, or how our immune system will react on these vaccines, and therefore we could develop these vaccines pretty fast. So what I'm trying to say is what they did is what we call basic science. So that is science we do just for knowledge, if you can say it in that way. There's no specific mission, just like with mine. When I'm going out and trying to detect a bacterial conversation, there's no specific mission in mind. But maybe in the future, we might have to look into these conversations because we want to communicate with bacteria. And then we need to have been started on this kind of research many years previously, because it takes 4 years to detect one compound. And I think it's so important that all of you guys is also part of that journey. So you also are able to understand what we're doing and also make some decisions when politics are occurring in that, or should we do this kind of agriculture or the other one, that you also know this is how research is working, this is how long time it takes, and we're all a part of it, and we all have to make decisions in it. Oh, that was such a long speech. I think that was it.